Hello, and welcome to the Membrane Structural and Functional Biology group. My name is Martin Caffrey. The focus of our research is the biological membrane, a schematic of which is shown on the first slide. The membrane, which surrounds the cell and subcellular organelles when present, is a molecularly thin structure just 2 lipid molecules across and studded with proteins. We are interested in structure and function as applied to both lipids and proteins. However, for purposes of this JoVE series, I will limit my focus to membrane proteins. We seek to understand how these membrane proteins function at a molecular level for 2 reasons. Firstly, there is the intellectual satisfaction in knowing how something works. Secondly, by knowing how it works, just like your bicycle or car, there is the prospect of fixing it if it malfunctions. Drug design is an obvious outcome of this type of work. The approach we take to figuring how a membrane protein works at a molecular level is to determine its crystallographic structure. This involves establishing the location in 3-dimensional space of all atoms, or at least all non-hydrogen atoms that make up the protein. The method we use for this purpose is called macromolecular X-ray crystallography, MX for short. Here we see an example of a membrane protein whose structure was determined using MX. A diffraction quality crystal of the protein is required to do MX. As you can imagine, there are many steps involved in structure determination using macromolecular crystallography as illustrated here. Typically, these include identifying a membrane protein target and then producing, purifying, and crystallizing it. Diffraction measurements are performed on the crystal using a home or a synchrotron X-ray source. The diffraction data are processed, yielding an electron density map, which is then fitted with a molecular model.